Did you know that this fish can swim as fast in water as a cheetah can run on land? Now anyways, in this video we're going to be continuing to talk about membrane transport. So we know that what is this thing right here? This is a cell and we're made up of billions of them, and each of them has a membrane, this little wall surrounding it. Not only is the membrane surrounding the cell, but there's also membranes surrounding organelles like the mitochondria and the nucleus. The main purpose of a membrane is to control what goes in and what goes out of a cell, right? That's the main purpose. Now, things can move in and out of a cell by various mechanisms, such as simple diffusion, facilitated diffusion, osmosis, and so many, all of these, right? These are ways by which things can move in and out of a cell. In the previous video, we talked about simple diffusion. Now, we're going to talk about osmosis. So every time I put a smiley face, it means we're either going to talk about it or we've already talked about it. So one key thing to note right now is that, remember, for simple diffusion, no protein was required. It just happened without this thing. It didn't need this thing. It just needed the phospholipid bilayer, and that's it. Now notice, for osmosis, we need this thing. We need this protein channel, and you'll see why. So let's just get straight into osmosis. So picture this. Here I have a little container. So yeah, just like a little container, we can see this water inside. Now, I separate the container with this wall. So what happens on this side is separate from what happens on this side. Okay, notice how each side is equal in the amount of water. But this side has a lot fewer molecules. So each of these squares here represents a solute, represents a molecule, right? So or a big substance, like whatever, any kind of substance. Now this side has way fewer of these molecules for the same amount of volume compared to this side. For the same amount of volume, this one has way more of these, right? Way more of these. Now, what will happen if we drill some holes in this wall, such as this? Let's say we have, let me move this up a bit. Let's say we drill some holes in this wall. Now, these substances are not allowed to move through this wall because these holes are too small, but the water can move. So these holes are big enough for water to move through, but this solute or this substance is unable to move. What do you suspect is going to happen? What's going to happen is water is going to move from this side through these holes to this side. Why is that? So this side has each of these molecules have a lot of water for themselves, right? Whereas each of these here have very little water for themselves because they're all sharing the same amount of water as these four molecules. So what's going to happen is this system, this container here, is going to try and reach equilibrium. This container is going to try and sort out the concentration difference. This side is way less concentrated, right? If you add a lot of sugar to your coffee or whatever, it's really concentrated with sugar. It's very full of it. Whereas if you add very little sugar, it's very dilute, not concentrated at all. Now, so we can say that this side is very concentrated with this solute, with this molecule, whereas this side is not concentrated. So once we open these holes, water is going to leave into this side to dilute this side. Because what's trying to happen is we're trying to reach an equilibrium. We're trying to even out the concentration between both sides. We're trying to make them equal. So water will move from this side to kind of even out this concentration gradient, make it more dilute until it's equal to this one. So it's kind of going to end up like this if you think about it. So what's going to happen is the water level on this side is going to sink and drop. Because it's going to start filling up this side to try and even out, because now this side is equal in concentration as this side. Each of the molecules on this side have access to equal amount of water than each molecule on this side. So that's what's going to happen. This is osmosis. Osmosis, in the most simple definition, is the movement of water from an area of low solute concentration to an area of high solute concentration in order to even out or reach an equilibrium that both sides have the same concentration. That's all that osmosis is. Osmosis is the movement of water, right, to even out things. So let's compare this to simple diffusion. Remember simple diffusion, we had a scenario like this. Say we have a similar scenario, so I'm making a simpler diagram here. Say we have this container and we have a separation in the middle, right? And each side has equal amount of water. Now let's say this barrier, this separation, is permeable to these solutes but not to water. What does this mean? This means this barrier can allow these solutes to pass but not water. So water can't pass, but these solutes can pass. What's going to happen in this scenario? Simple diffusion. These molecules will move, some of them will move from the area that is more concentrated to the area that is less concentrated to even out the concentration on both sides. So basically this is going to happen. If we have this image, one of these molecules will move to this side because now this side is equal concentration to this side. We're trying to reach an equilibrium. That is simple diffusion. Now let's say we have another scenario here where this barrier cannot, unlike this barrier here which allowed these solutes to pass, this barrier cannot allow these solutes to pass. These solutes or these molecules cannot pass this barrier, but the water can. So how are we going to even out these concentration gradient differences? This side is way less concentrated than this side. So how are we going to even it out? Because in the simple diffusion example, the molecule just moved to the other side and now it's equal. But now I'm telling you that this barrier cannot allow this molecule to pass. It can only allow water to move. So how are we going to sort out this problem? How are we going to reach an equilibrium? By osmosis, what I just explained. If this barrier is permeable to water, that means the water will go from where there's very few molecules, because these two molecules have so much water for themselves, whereas these four are sharing all of this water. So the only way to make this fair is if these two molecules lose some water and give these molecules more, because then they all will have equal overall. So basically what's going to happen is this. Water molecules will move from this side to this side. Now, if we think about it, each of these four molecules have their own square of water, their own area of water, just like these two. So it's now even. We've now reached an equilibrium. Both sides now are equally concentrated. This side has is as concentrated as this side. That's the same thing that happened here with simple diffusion. This side is as concentrated as this side now. So you can see the difference between simple diffusion and osmosis is the similarity first is that both processes are trying to even out or reach an equilibrium between both sides to even out their concentration. Simple diffusion does it through the movement of molecules, so these substances, whereas osmosis does it through the movement of water. So if we had to put a definition here of simple diffusion, okay, we can say it's the movement of substances down a concentration gradient, right? It was very concentrated here, very low concentration here, so it moved from the area of high concentration to the area of low concentration in order to make this equilibrium. That's what simple diffusion is. Now, to define osmosis, it's very similar, but there's some slight changes in the word. I'll just go straight to the key definition. It's the movement of water from an area of low solid concentration to an area of high, to even them out, to make them equal in concentration. So in reality, that's the difference. So osmosis doesn't have, so if we think about a cell, so let's think about a cell, we know a cell likes to bring things in and out. So osmosis is the process by which a cell brings in water or brings out water. So no substances move by osmosis, whereas simple diffusion can bring substances in and substances out, like oxygen or carbon dioxide, like we talked about in this video. So one last thing we need to do here before we end this video is very important. So I have here 3 scenarios for you, and we're still thinking about osmosis. So we have 3 scenarios. I have one scenario here where this is a cell, by the way. I made a very simple cell, so there's no organelles. We're just caring about osmosis here, no organelles required. Here's the nucleus, and we have this membrane, right? This membrane. So to make this process of osmosis relevant to the human body, because so far we just talked about this setup. We didn't talk about the actual cells of the human body and why osmosis, how it works in our body. It's the same thing, but I'm just going to show you here. So if we have these 3 scenarios, what's going to happen in each of these scenarios? So in this scenario, we have equal the inside is equal in concentration to the outside. Okay, so what's going to happen here? Now this scenario here has a very high concentration of this substance. So this is just a substance, this little image here. It's just like a molecule, basically. Think of it like a molecule. So in this picture, this inside of the cell, the cytoplasm, is very concentrated, whereas the outside is very dilute. And then this last scenario here, the inside of the cell is very dilute and the outside is very concentrated. What do you think is going to happen in each of these scenarios? So let me show you. So what's going to happen is that nothing is going to happen in this one, right? Because if the inside is equal in concentration to the outside, nothing will move, because as soon as water moves or anything, then we're going to disturb the equilibrium. That's pointless. Right now it is in equilibrium. It is fine. So nothing is going to happen, right? So you can think of it like a water bottle. It's not going to expand, it's not going to shrink, it's going to stay just normal. We call this process, by the way, when the cell is when the inside is equally concentrated to the outside, we call this, let me bring this to the front, we call this isotonic. So this cell, this is a key word you need to know. This cell is isotonic. Iso means equal, whereas tonic means water pressure. So both the inside of the cell and the outside have equal water pressure. Okay, equal water pressure, because both of them have the equal concentration. So now, what about this scenario? So here it's super concentrated, where the outside is super dilute. We want to make it equally concentrated. So what's going to happen here? Osmosis. Water is going to move from the area that's very dilute to try and bring some water from where it's dilute to an area where it's concentrated, to dilute this area and make it more equal to the outside. So essentially, this will happen. We will start expanding this cell. We will start, just pretend all the molecules are in here. Well, I can move it, it's fine. So you can think, oh crap, what was that? So you can think that now, because water moved in and these molecules couldn't go anywhere, now we have a super swollen cell. Oh, this is not working. Okay, super swollen cell, because now the cell is, the equilibrium has been reached. The cell has been diluted. Now the cell is equal concentration to the outside. Okay, so you can think of this as a bottle that was filled up so much it started expanding. It's about to pop. So this is bad in real life because your cells can eventually burst and die. So how does this scenario happen? If you think about this, the outside of the cell has a lot of water. How does this happen? When you drink too much water, if you drink way too much water, you keep drinking and keep drinking, you end up making the area right outside of the cells so dilute that when osmosis happens, the cells burst. So you can literally kill yourself by drinking too much water. Now, what do we call this kind of scenario here? We call it, this cell we call hypotonic. Hypo meaning low, and tonic meaning concentration of water. So in here, we have a low concentration of water. We have a high concentration of solute, a low concentration of water. So this was an isotonic, this is a hypotonic. And in a hypotonic cell, it will start expanding when osmosis happens, which is bad, right? We don't want this to happen. Now, last scenario here is reverse. So we can suspect that water will in fact leave the cell to dilute the outside environment, right? That's exactly. So the cell will now start shriveling up because it's losing all of its water. It's like filling up a balloon with water and then squeezing out all the water. The balloon becomes shriveled up, right? So that's the same with the cell. After the water leaves to dilute the outside, the cell will end up looking like this, all shriveled up like a bottle like this. You suck out all the air, basically. Now, what do we call this kind of cell? We call this kind of process here that this cell was hypertonic. There was a lot of water pressure inside here, not a lot of solutes. So in a hypertonic cell, the water will leave. There's too much water in here compared to the outside. So these are three very key words you need to know. And again, this can happen during dehydration. If you're starving, you didn't drink water in a long time, the amount of water outside of your cells is very low because you're not bringing in much water to supply your cells. So the water that was supposed to go to your cells, it's very low here. So water is going to end up leaving your cell by osmosis and they will shrivel. And this is also very bad, just like a hypotonic scenario. So these are two ways which are very bad. You don't want this to happen necessarily in a human body, at least. So what's one other meaningful purpose of osmosis, of knowing how this stuff works? It's in transplantation. You know, sometimes you got to move your organ from one body to another body if you want to like save a person. The second you take this organ out, think about it. It's made of cells. So you don't directly put it into a new person. You first put it in a container to prepare the body that it's going to go to. Now, when you put it in water, you got to make sure that the water you put it in is isotonic. Isotonic. So when you take out this organ and you put it in water, you got to make sure the environment is isotonic so that this organ, which is made up of cells, that the cells do not stretch or shrivel up, right? You want to make sure the environment is isotonic so that the organ can remain in good shape. Because if you don't ensure the isotonic environment, then one of these two things will happen and the organ will die before it can even get to the new recipient. So that's one other very important reason to understand this process of osmosis. Now that's it for osmosis. So in one of the next videos, we will continue and talk about the rest of the stuff here.